I hope my screen is visible to you, right? So in the last class, we discussed a few things, which included about the mechanisms of replication. In that, we have seen about the DNA polymerase or the holoenzyme, right? This is the sliding clamp that we have here, and we can also have, this is the sliding clamp loader, and there are at any given time 2 DNA polymerases that will replicate, but there are actually 3 DNA polymerases attached. One, and this is 2, and this is the third one. So for lagging strand, you need 2 different DNA polymerases, one after one. They will work to polymerize the lagging strand discontinuously. Whereas polymerase, the other one that is working on the leading strand, will keep on polymerizing it continuously. So this is a complex mechanism. I think yesterday I tried to show you a video where the visualization is better, right? Quickly though, we can look at these things. Look at some of the stuff that we have discussed in the last class. It's all about trying to manage leading strand and lagging strand in the same direction, or the technical problem of direction of replication and direction of polymerization should be in the same. So for that, how does it solve that problem is what we have discussed. I think it's better observed when we are doing it, when we watch the animation. So I just want to remember that there are 4 different things that we will ask in exam. One is chemistry, and second, the mechanisms of polymerization, which will include, the chemistry of polymerization. We should be able to talk about the 3' OH making an attack on the alpha phosphate and then making a phosphoroester bond. We should also be able to talk about base pairing and many other aspects, including metal ion A and B and so on. So how the pyrophosphate is released and what happens to pyrophosphate, and several things like that. In enzymology, you should be able to talk about all the protein components, including RNA primer, that are involved in the process of polymerization or replication. So something like single strand binding proteins, helicase, you can talk about topoisomerases. All those things will come in the enzymology part. In mechanism, how the holoenzyme and how exactly the mechanism happens is what you will talk about in mechanism. And then we have something about regulation of replication, but we also have one important thing that we have to discuss is about topoisomerase. Okay, and in that we will also discuss about end replication problem. So we discussed about the origin of replication and how initiation happens. Yes. So we discussed in the last class about the uh, replicator and initiator model, right? That is what is given here, the replicon model, which has a replicator and an initiator that recognizes the replicator, eventually resulting in 2 different DNA molecules. Mostly we are talking in circular because all bacterial chromosomes are circular. All eukaryotic chromosomes um, are numerous, and then they are linear. So in bacterial chromosomes, typically bacteria have one circular chromosome. Okay, but some of them have 2 circular chromosomes, like Vibrio cholera and other bacteria. They have 2 circular chromosomes. Whereas in all eukaryotes, you will find that there are linear chromosomes, and there are usually multiple in number. So this is um, about the replicators. There are 9 mers, several sequences in this way, and 1 second. Yes. And then there are 30mers, that is in E. coli. You call it as ORIC for origin of replication. So there are three proteins that are involved in initiation of replication in E. coli. That is Escherichia coli, DNA A, DNA B, and DNA C, or their common name would be DNA A is called as such, whereas the DNA B is helicase and DNA C is helicase loader. So DNA A protein binds to the 9-mer sequences repeats that are present here, and then they kind of twist the DNA so that the DNA that is present in this 13-mer usually opens up. And this 13-mer will have kind of reduced or increased AT content, AT, instead of GC. You will not find so much GC in it because the region of the DNA where we are expecting that the denaturation would have should be rich in AT, because it will be less number of hydrogen bonds, so it's easier to denature. So the binding of DNA A will induce a denaturation process in the 13-mer region, like in here, and then the DNA is denatured, and then DNA B and C, as a complex together, they will come, and the loader, helicase loader, will put the helicase onto the one of the strands. And helicase is a active or it is denaturation of DNA is an active process, which means that there is uptake of or consumption of energy, and it moves. The helicase is surrounds one of the DNA molecule as shown here, one strand, and then it moves from 5 prime to 3 prime direction, like this. Okay, and then helicase will keep on moving. So this one is showing, yeah, and as they keep moving, they will keep denaturing more DNA, and then the DNA polymerase, that is the holo enzyme, DNA polymerase 3 holo enzyme, which consists of the tau protein, helicase loader, and so on. Sorry, the sliding clamp loader and clamp, they will organize something like this. And then we have the sliding clamp binding, and then the leading strand will start being synthesized, and then the lagging strand will also be started to synthesize. So what we are going to learn is now is something crucial about when should the replication happen. Do you want it to happen? It should be orchestrated. Say a bacterium is like this, and it will undergo something like a mitosis to give rise to two different molecules like this, right? And in that case, the chromosome that is present in the cell should also time its replication so that it will produce two different DNA like this. If there is one cell and the replication did not happen, and the cytokinesis happens, just a theoretical one, then one of the daughter cells will get the DNA and the other daughter cell will not get the DNA, and therefore it is going to die. It's useless product, isn't it? That's all the consumption of energy growth would have been wasted resources. So there should be a timing of DNA replication, and that should go hand in hand with the DNA with the um, cell division. And you can also think of it this way: if there is a cell, it is not having nutrition, or it is not getting enough energy or matter, what would you expect this cell to? Should this cell undergo cell division or not? If there is no input of energy and matter, then the cell cannot grow. And if the cell initiates a process of cell division, then the cell would die, right? Because there is incomplete everything. And at the same time, replication should not have occurred because it's consumption of energy. So, and which is in short supply at this stage. So that the regulation the replication should be highly regulated and should happen only at stipulated times so that there is no problem in the sense that there is um, it does not impose any problem onto the cell. So here is what we will do. We will try to learn this concept. It's important. The first thing is that there are now we are trying to learn finer details which will get complex. So please try to pay attention and try to read your textbook as well. In E. coli, at least, there are several sequences like this, GATC, right? So that are shown here. I will just take this up and then we will discuss. So this is the 5 prime and here is 3 prime. The complementary strand is anti-parallel 5 prime to 3 prime. Now if I have to read the sequence on the bottom strand, you can say I have to read always from the 5 prime side, G, A, T, C, right? You read it this way. On the top strand, G, A, T, C. Read it on the bottom strand in this direction, G, A, T, C. Either say you're reading the same sequence. It's always G, A, T, C. Anything like that is called as a palindrome. You must have heard in some cases at least. Palindrome, right? The ones that you're reading from here to here or here to here. I am emphasizing on this because when you read about restriction endonucleases, you will find several sequences that are like this. So there is a protein called DAM methylase. Okay, it is an enzyme. And what does it say by the name of it, at least this part? It is a methylating enzyme. And what does it mean? It means it methylates adenine. So you say GATC and here GATC, right? So this particular protein has something, it is sequence specific. That means it only recognizes GATC sequences. And once it recognizes or comes across a sequence like that, GATC, it will methylate the adenine, something like this. Okay, it methylates on the N, that is amino group present on the 6th carbon of adenine. I hope you remember there is adenine is 6-aminopurine, and on that amino group that is present on the 6th carbon, adenine methylates that region. So GATC, you have it. So the important thing about the enzyme that makes it is this. And also try to figure out the terminology that we are often using, right? Because it's important that you speak. I say it is sequence specific. What does that mean? It will methylate adenines only in these regions, where in this sequence, GATC. Okay, there is a high probability of GATC occurrence all over the genome of E. coli. And everywhere this enzyme just keeps going around and sequence and then methylating the adenine molecule, adenine in that place. So here is the DNA molecule here. I will use it. So this is the DNA molecule that you are seeing. Because dimethyl is produced, all the GATC sequences are usually methylated like this on both the sides, here and here. Okay. Then if initiation of replication happens, new DNA is being synthesized. This is the new DNA, new strand that is being synthesized. This is the parent strand, right? These are the parent strands and these are the new strands. I mean, it is semi-conservative replication. I hope you remember that, right? In semi-conservative replication, one comes from the parent strand, one is parent strand, the other one is newly synthesized. So it's the same case with the bottom DNA molecule like this. One is old, one is newly synthesized. The old one already has the methylation here like this. So that strand will contain all the methyl groups. Whereas the newly synthesized strand is not having any methylation. The same is the case here also. New strands do not have any methylation yet. Okay, that is called as hemimethylation. The term for that is hemimethylation. What do you think? What do you suppose would be the difference between hemimethylation and semimethylation? I am telling this only for the sake of trying to understand what is, how you can probably remember what is meant by hemimethylation. Say, for example, if there is a DNA molecule, something like this, and assume that each of the the blue circles that I'm putting are GATC sequences. Okay, and if there are certain regions wherein you have this, there'll be two A's, one on top strand, one on the bottom strand. So is the case with all. So this is a completely methylated state. You call that simply as methylated DNA. Say, for example, I have a situation like this. One second. Okay, you have seen the methylated state. Now I would want to tell you, I want you to tell me what would happen here. What is this? Okay, something like this. And there is another similar state where I will draw. These are the same things I'm trying to put. The methylation pattern is only different. Okay, let me put all the three so that you will be able to distinguish what it is. And say, molecular biology, like any of the biology, will require a lot of memory, right? You have to memorize a lot of stuff, and then there is too much of things to memorize. In that case, one should try to associate, try to learn the term properly. And here, that is what I'm trying to do. What is hemimethylation? So the one that I drawn here, it is methylated state, completely methylated one. You'll just call it as methylated DNA. If there is a DNA strand like this with the DNA molecule with methylation pattern like this, you can probably call it as semi-methylation. It doesn't have so much of significance in our biology. But if you have a DNA molecule like this, right? All one of the strand is methylated, whereas the other strand is not methylated, that you call it as hemimethylated. And that is what you are seeing in this place as well. That is why you need to, that is how you try to remember the terms so that you don't have to remember the whole of everything else, right? Methylation is on both sides, the DNA is methylated, not everywhere, because dimethylase is specific, sequence specific, meaning that it only methylates the adenine that is present in sequence GATC. Okay, so there are several, there is a protein called SEC-A. SEC-A is a sequence specific DNA binding protein. When I say that, what does it mean? It by the term itself, it means that it binds to DNA. First thing is it binds to DNA. It doesn't bind just randomly anywhere in the DNA. It binds to only certain sequences, and those sequences are GATC. So SEC-A binds only to GATC sequences. There is one more stimulation to it. It only binds to DNA sequences that are hemimethylated. This is hemimethylated. If there is a DNA molecule which has GATC and only one of the adenine is methylated, then SEC-A goes and binds there, like as shown here. Okay? And so if it is methylated like this, I hope you understand that when I say methylase is a methylase, it will methylate DNA in a sequence specific manner and at GATC sequences. Methylase can come here and methylate it. But there is the obstruction of SEC-A. SEC-A is a protein it binds to hemimethylated DNA. And where does it bind? It binds exactly in the same sequence as GATC. It is also sequence specific. So if SEC is bound here, DAM methylase cannot come and methylate this DNA, this new strand, isn't it? So there is a delay. SEC delays the methylation of the new strands, because of which there is no possibility for renewal or reinitiation of that replication. Okay, so I hope you understood this part of it. So DNA methylase, SEC binds and then will kind of delay the methylation of the second strand. If the second strand is also synthesized, SEC does not bind. SEC binds only to the hemimethylated GATC sequences. So often what may happen is SEC might dissociate. And when SEC dissociates, then DAM methylase may have an opportunity to methylate the new strand as well, something like this. Okay? And when that happens, SEC cannot bind here any longer. So gradually, more and more DNA becomes hemimethylated, and then SEC is no longer bound here, and then DNA A, the one that binds to the 9 mers as shown here, will bind here and will denature the regions of this region, something 13 mers, and reinitiate next round of replication. So this is not a complex mechanism. When we try to learn about newer things, something like eukaryotic ones, it becomes complex. I will try to discuss that in the next class. And but I want you to read the textbook, watch the videos. I hope you have watched the cell cycle video. Please do that, and also that of replication, and come for the next class. In today's class, we will want to discuss about okay, eukaryotic regulation of replication. We will have a look at it in the next class. Today's class, my focus will be on telomerases and the end replication problem. I think it's not here, but I will try to use some space. Just give me a second. No. Okay. Yes. So in this class, we will try to discuss something about end replication problem. We told that DNA polymerase has a problem, isn't it? What is DNA problem? DNA polymerase, an issue that is associated with it. It cannot synthesize, it cannot initiate DNA replication by itself. It always requires a 3' OH. And the substrate is this primer template junction. So the top one, you can call it as the primer, and this one as the template. And DNA polymerase can only add more and more bases to it, something like this, right? That is the origin. That is the problem with DNA polymerase. Why do I say it as a problem? Let me draw it a little clearly. So here is a DNA molecule. Assume that's the template, and now I'm going to draw a primer, and that is the primer is this. 1 second. This is the primer, and then assume the DNA polymerase has synthesized the or polymerized the next bit of the DNA. I'm just putting it for only one strand I'm showing. So eventually RNase H or DNA polymerase 1 should come and delete the or remove this primer, right? That is what they will do. Now we have a gap, and what to do with this gap? How to fill this gap? DNA polymerase cannot do it because there is no 3' OH. And so there is no way of doing that. So assume that these two are now undergoing second round of replication. I will try to put what will happen for each of the cases. So assume they are of the same length. Sorry, let me put. This is what will happen for the lower strand, and you will have the newly synthesized strand for this one. So I'm talking about for this strand, okay? And you also have new RNA, that is RNA primer, right? It requires a RNA primer. What will happen to the top strand? I'll try to put it here. Top strand is already little smaller, isn't it? And so I will draw it as little smaller. Assume that I'm doing it on the other side or so, but I'll put it here for ease of understanding. So there is a primer, and then we have the DNA strand like this. Now what should happen? We would want to eliminate the primer. After one round of replication, what has happened? It has become shorter. This is the first round you can think of, and this is the second round. And if this keeps on going, what is going to happen? The genome or the chromosome will become smaller and smaller after each round. Okay, if you watch the animation, you should have a better understanding of it. And that is called as end replication problem. Right? Any linear DNA molecules will have this issue. So why did I say linear molecule? I should have started with that. If it were circular DNA, like this, something like this, there is no problem, because it will denature the DNA, something like this, and it will form two replication forks. This is the new DNA that is being synthesized, assume like that, right? One replication fork is moving this direction, the another one is moving in this direction. Eventually they will form, they will meet at this place, and they will just form two circles that are interlinked, something like this. And sorry, they are interlinked like this, which will be resolved by topoisomerases, leading to formation of two separate or independent DNA molecules like this. So for circular DNA molecules, there is no end replication problem. Whereas for linear ones, there is always a problem, something like this, because the ends of the DNA molecule cannot be synthesized by DNA polymerase. Why can DNA polymerase do that? Because it has a dependency on 3' OH. It cannot by itself initiate replication, right? It cannot initiate polymerization. It needs a 3' OH. There is a obligate dependency for it. Then how is it resolved is what we want to talk about. So in that case, we should discuss about topo. No, I will just look into the animation. If it is around, we should just play that. Telomerase. I'll just play it in few minutes. Just wait there. And there we will also have a discussion about end replication problem. Try to have yes. So This is a problem. We will put it as this. 1 second. I have to get used to the this open board clearly. So we have, this is the DNA strand, and the other one I will put it as this. Okay. If I label, you will probably understand what I'm trying to mean. So this is the 3' side of this. This is 5', and here is 3', and here is 5'. And here is the gap that needs to be filled. Assume this is the whole gap that needs to be filled. I'll just put it in this bracket like this. And why can't DNA polymerase fill that gap? Because it cannot fill it, because if it has to fill, there should be a 3' OH here. Only then it can fill up like this. Otherwise, it cannot. So there is something called as an enzyme. It's a ribonucleoprotein. Ribonucleoprotein. What does it say? That means it has two components. That is, it has an RNA component and it has a protein component. And that is, the enzyme name is telomerase. That means it synthesizes the telomeres. The ends of these linear DNA molecules, typically in eukaryotes, they are called as telomeres. The enzyme that produces them are called as telomerases. Telomerase is a ribonucleoprotein. It has RNA component and it has a protein component. When I described DNA polymerase, I told you a way to remember it. I don't know if you can remember that or not. DNA polymerase is a DNA dependent DNA polymerase. What does that say? This is the whole definition of DNA polymerase, which is DNA dependent DNA polymerase. It polymerizes DNA, and it is all, the template must be a DNA. This is, dependency is about template. What is the template in replication? It is DNA. And what is the thing that is formed in polymerization of DNA? It is DNA polymerase, right? What is the being formed is what you're calling here. In telomerase, why I discuss this DNA polymerase or trying to recollect it is, you can think of telomerase is different. I will try to write it and you can memorize it that way. RNA dependent DNA polymerase, right? Then if I said that, what should be the template? It should be, RNA should be the template. And what is it going to make? It is going to make a DNA strand. The newly synthesized one is going to be a DNA strand. So telomerase, as I said, has two components. One is RNA and the other one is protein. The RNA component acts as a template. And protein, along with other RNA components, will act as DNA polymerase. I hope you heard the term called reverse transcriptase. Right? Reverse transcriptase means, from, if anybody is in general, if you are producing DNA from RNA, you would call it as transcription. And the enzyme is RNA polymerase. It is producing an RNA transcript from DNA. If somebody is producing from RNA, you're producing a DNA. It is like a reverse, it is going in reverse direction. And transcriptase, it's something like producing a DNA out of RNA. Okay? So the mechanism is beautiful actually about how DNA polymerase actually works. We'll just try to get this again done. I hope you understood that DNA polymerase, sorry, telomerase is the one that synthesizes telomeres. It is a ribonucleoprotein. It has a RNA component. It has a protein component. And telomerase is a RNA-dependent DNA polymerase. RNA component acts as a template, and telomerase synthesizes DNA strand. That is something you need to remember. So I'll use this again. So you have this part, and here is this. We have a problem, and the problem is that we are unable to synthesize. This is 5 prime, and this is 3 prime. We are unable to synthesize this region of the space that is here. We are unable to synthesize this region. So what DNA, um, um, what it does is, give me a second. Okay. What the telomerase does is something like this. Telomerase acts as a template where you will have it. If there is a sequence, it will bind something here. Telomerase RNA component may bind. Oh, sorry. Yeah. DNA RNA polymerase will bind here. The RNA component of telomerase, sorry, will be bound here like this. This is going to act as template, this region. So what does the rest of the telomerase do? It will synthesize DNA molecule out of it like this. I'll put it in red so that you will understand it as DNA. What does telomerase do again? It will move one step here. And that means what is going to happen? Let me copy paste, see if I can make it up. No. Okay, I'll just keep drawing. Assume that this is the region that is being synthesized. The next step would be translocation. The telomerase that was present in this place will move something like this. And then it will also synthesize more DNA molecule. It is just extending this region by a large margin. So it will now move to the next position like this. And then more DNA molecule would have been produced, something like this. I hope you see what is happening, what telomerase is doing. Telomerase is extending the lower strand. Why is it extending? What is the use of it? Because this gap is not yet filled. The idea is that we want this gap to be filled, right? And if that gap has to be filled and you're extending this the lower strand, what's the use of it? The use is that now you have a large amount of DNA on the bottom strand here. So what can um, primase do? It can come and synthesize a primer here. That means primer is for to supply the 3' OH, which means that DNA polymerase can now take over this and synthesize the new strand, this part. What is the objective? To fill this gap. And did nature accomplish it? Yes, it did. It has filled up this gap. How did it do? It has extended the lower strand by a large margin. It has extended this a long way so that a new primer is synthesized, using which the rest of the DNA is synthesized. Right? Why is this so important? I hope you will realize that when we study in the next class also, you will know that the DNA, the genome, right, or the chromosome is something like this. It has numerous genes all over, right? The ends also may be having a gene, something like this. If if it is not for the telomerase, what would have happened? The chromosome would have become shorter and shorter, and it would have lost some of the genes that are in the periphery like this. And if some genes are lost, the cell is dead, right? Therefore, you can see that what telomerase does is it keeps extending this so that the end replication problem is solved. By extending it, you have a primer that is produced, and then you have this you have the missing parts being synthesized so that the chromosomes don't become shorter. Yes, I hope we will try to watch the video now, and I hope you will get the best out of it. And then if there are any questions, we will discuss on that. So that's the DNA polymerase. It is synthesizing, and that is how the looping of the lagging strand happens. So what you will learn from this particular thing, I mean, I will try to recollect while they are saying is you should be able to understand that telomerase is a ribonucleoprotein, and it has RNA component, and it has a DNA component and a protein component. Yes, Okazaki fragments, RNase H, they are removed and the gaps are filled. As long as the 3' OH is there, it can be filled by DNA polymerases and ligated by ligase. So when the chromosome is coming to the end, as shown here, what will happen? You keep observing it. You can see that there is a primer that has been synthesized in this one, in this strand, and the new DNA strand has been made. RNase H removes this part, and the is filled, ligase fixes it. Now RNase H removed it, and now what to do with this? This is the unreplicated single strand DNA. That's the gap. If replication continues further rounds, slowly the chromosome will become smaller and smaller, as you are seeing here. And see, this one has become smaller, and for the next round, it becomes even further smaller, and eventually there will be a loss of cells. But luckily, we have something called as telomerases at the end of our DNA, and those telomerase, we will also discuss this in the next class at the sequence level. I don't want you to get too much into it, but try to follow this video properly. It extends this region by a large margin. Let's see. This is the telomerase. This is the RNA component. This region of the RNA component acts as the template. So it is also RNA dependent DNA polymerase. It's a kind of reverse transcriptase. So the template is complementary to the telomerase region. I want you to also appreciate the structure of the template. AAU, CCC, AAU. I don't want you to mug up the sequence. I want you to see the design of the sequence. AAU, C, and CCC, AAU. Yeah. So it slowly synthesizes the complementary strand. And what does it do? Next step is called as translocation. It lifts up and move further. See how is it moving? Something like this. It binds here. And now it synthesizes this part. Again, translocates. It's a beautiful mechanism. And the beauty, I think, is two-way. First is nature is beautiful for having come up with something ingenious or intellectual ways. But even more astonishing is the scientists who figured it out. Right? It must have taken millions of years to come up with something like this, and human brains, many of them together, could come up with uh, figure out how things have happened. The beauty of when you're doing science is the best part is there are two way. Basic science is trying to find out what is there. Applied science is trying to use what you found. I think as a scientist, everybody should start as a basic scientist because you don't know what is there and it is full of uncertainty and you are trying to figure it out. Then you should be in that situation and try to figure it out. That is where you learn to deal with uncertainty. The applied science is about trying to use what you want. You have an objective to reach, that is, you want to find a solution for a problem, and then you should use any means to get the problem solved. So, these are the two skills as researchers or scientists one has to employ. And if you look at biology as if it is intelligent, you will find so many logics all over about how to make things happen. And imagine that all these things have happened by themselves. That means it has come about by through natural selection. Those that were able to be stable will survive. Those that are not stable will die out or go extinct. Okay? So we have 1 minute. If you have any questions, please. I hope you understood telomerase. In next class, I will take up telomerase again. We will deal with sequence level on the board, and then we will see what else we have to do. Eukaryotic replication and other things are there. I'll stop here. If there are any questions, I'll try to address them.